Okay, hello people. So this is video number 2 in our carbohydrate catabolism series. Last time we did glycolysis and fermentation, and this time we're carrying forward to finish out the catabolism or complete oxidation of a glucose molecule. So after watching this video, you should be able to state the eukaryotic cellular locations for PDC and TCA, draw the substrates and products for the PDC, correlate coenzymes with subunits and the vitamins, draw the TCA pathway with names of all 8 intermediates and corresponding number of carbons. Notice that you don't need to know the structures of these. Add in CO2, NADH, FADH2, GTP, and CoA to the appropriate steps as they come out of the TCA cycle. Some of them go out and in, like CoA. State the 3 irreversible enzymes in the TCA cycle. Describe the thermodynamics of synthesis and hydrolysis of a thioester bond. Write the overall chemical equation for the PDC and recognize the structure of coenzymes in the PDC. Last time we had taken glucose, which is a 6-carbon molecule, and partially oxidized it to 2 pyruvates. And that turns out it releases about negative 146 kilojoules per mole. But the complete oxidation of glucose, where all, so that should be a 6 O2s, where all 6 carbons in glucose go to 6 carbons in CO2, and we also get out 6 waters, just so you know, this part right here is electron transport chain. This part right here is glycolysis plus the PDC plus TCA cycle. Hopefully those don't all just look like letters. But this right here, if the complete oxidation of glucose, so we turn all of the carbon-hydrogen bonds into carbon-oxygen bonds, releases 2,900 kilojoules per mole of energy. So what I'm trying to say is that we have a long ways to go. And glycolysis is definitely not the end of the story here. We need to take these, all 3 carbons in pyruvate, need to eventually get to CO2. And that's where we're going to go. Okay, so next we're going to oxidize the pyruvate from glycolysis all the way to CO2. I just did that. And this is what the PDC, or sometimes this is referred to as the bridge step, because it bridges glycolysis and the citric acid cycle. I'll refer to it as PDC, or PDC stands for pyruvate dehydrogenase complex. Sometimes it's also referred to as PDH, same thing. The citric acid cycle is also called the Krebs cycle. It's called the tricarboxylic acid cycle, and sometimes it's called the TCA cycle. I think I probably use TCA cycle and citric acid cycle the most. Just food for thought. Okay, so where do these occur? In a eukaryotic cell, these both occur in the mitochondrial matrix. So here's a eukaryote. You know it's a eukaryote because it has a true nucleus. And there's a whole bunch of other organelles. This little pink one right here is the same as that. And so what you'll notice is that the outer sort of shell of this is a membrane. And then it has a second membrane on the inside. And there's a sort of watery, liquidy area between, that's all the way on the very, very, very inside. And that's called the matrix. This is kind of equivalent to the cytosol, and just in the fact that it's the watery, liquidy area. But you'll find both the PDC and the TCA cycle hanging out inside the mitochondrial matrix. Okay, so we're going to start with the PDC first. So where we ended was we made some pyruvate. Here's pyruvate. Please remember how to draw this. And the overall reaction for the PDC is that there will be 1 pyruvate going in, an NAD+, and a a CoA molecule. And what comes out will be an acetyl-CoA. Acetyl means there's just a methyl group here. Acetyl. Acyl, A-C-Y-L, it means there's many, many carbons coming off of the carbonyl. 1 CO2 and an NADH. So everything is accounted for here. There's a few things that I want to point out. Pyruvate is a 3-carbon molecule. There's 1, 2, 3. If 3 carbons go in, 3 carbons must come out. Hopefully you might be able to see that this is already a CO2, so that was what carbon number 1 is. And then here's carbon number 2, still the carbonyl, and carbon number 3 is the methyl group. Okay, so we've made our very first CO2, which is great, and we have 2 more to make eventually, and those will come out in the TCA cycle because there's 2 carbons left. So like I said, the PDC is a bridge step on the way to complete oxidation because we do get 1 CO2 out, and it does create a molecule that goes on to be completely oxidized. Alright, so the PDC tends to make people's heads kind of spin around and around and around because it's a ginormous enzyme, and it has many, many subunits. And the reaction that it catalyzes in each of the unique subunits, there's 3 unique subunits. And then there's just repeating ones of those, and those are called, the repeating subunits are called E1, E2, and E3. And each of them has a unique or several unique. So this one has, E1 has the coenzyme that we abbreviate as TPP. E2 has a coenzyme, lipoamide and CoA, and then E3 has NADH and FADH2. Okay, and so that's 1, 2, 3, 4, 5 coenzymes, almost 2 per subunit, so there's a lot going on, and we're going to draw this. You do not need to memorize the drawing of this mechanism, but I don't know how else to introduce it to you. Talking through other diagrams doesn't seem to work very well. So we're going to just draw it. You'll just need to recognize the structures of all of these. You'll see in some of your assignments I have you fill in the blank, so don't panic on the drawing part. But we're going to do it just so that you can at least see it one time, because it's basically like a giant assembly line enzyme, and it's kind of cool, but it does make people's heads spin just a little bit. Here we go. Alright, so we're going to start with pyruvate. Again, one of those molecules that you need to know its structure. 3 carbons. 1, 2, and 3. The first thing that happens is carbon number 1 is decarboxylated. Whoops, hey, go away. So carbon number 1 is going to leave. That's our first CO2, so a complete oxidation of that carbon. The other 2 carbons are going to get passed on to what's called a thiamine pyrophosphate. It has a really cool structure. Again, you don't need to memorize the structure of it. I'm just doing this so that you have seen it at least once. So that carbon is a carb anion. We have a positively charged nitrogen. There's a sulfur in there. It's like almost everybody gets a shot here. Okay, so this is called thiamine pyrophosphate, and we abbreviate it as TPP. Okay, so the negative charge on the TPP is going to be the one that's going to attack here. So we'll get a CO2, we'll leave, and then this whole thiamine pyrophosphate, which is part of, if you remember, it's part of the E1 subunit of the PDC complex, yikes, is going to grab a hold of those last 2 carbons. And so what it looks like is we've got a carbon, attached to a carbon. Those electrons here will go up and snatch themselves a hydrogen from water. And then again, we'll have the methyl group. So if I'm keeping track of my carbons, this is carbon number 2 now, and this is carbon number 3 from the original pyruvate. And then, can you hear all of that? Yeah, can you tell that I'm not at home? I'm just going to pause it. Okay, I hope that whoever they're going to, destination-wise, I hope they're okay. Alright, so here we go. Still just drawing. I guess I could have just carried on with that. Hey, I don't want to do that. Hey, don't do that. R group here, R group here. Okay, so now we have the 2 carbons that have been passed to a TPP, and then they're going to get passed again. So we're going to regenerate our original thiamine pyrophosphate. And in the process, a lipoamide is going to come and grab those 2 carbons. And I'm actually not going to draw this one out because it doesn't make as much sense as the other one. But those 2 carbons are going to be passed on to this lipoamide. Also, it kind of looks like a little house here. Put these guys back in blue. We regenerate the carbonyl. We have our acetyl group. And again, this is carbon number 2 and carbon number 3. And this funky looking thing, whoops, I put that in the wrong spot. This R group right here, this is called lipoamide. Okay, it's also called the swinging arm, and I'll show you what that means in just a second. Okay, this is all happening at E2, and I'm going to scrunch this and move it over just a little bit. There we go. This is all happening at E2, and another thing that happens at E2 is that the CoA comes in. There we go, SH-CoA, and this SH group will come, attack the carbonyl carbon, electrons go up, down, and then it will leave the lipoamide. So the lipoamide will not be fully regenerated yet, that's an S, but what's coming out is we have our S-CoA, that's our CoA, and then that is attached to the acetyl group, and we have formed acetyl-CoA, which is a second one of our products. So if you remember, we have 3 products. We'll have CO2, acetyl-CoA, and we'll have an NADH. In order to reform lipoamide, these hydrogens right here, they have to go. And so they go on to, first, they go on to an FAD, which gets reduced to FADH2. So those 2 hydrogens end up as those 2 hydrogens. And then in order to regenerate FADH2 in the E3 subunit, those hydrogens are going to get passed again from an NAD plus onto an NADH. Yikes. Okay, so there's product number 1. Here's product number 2. And here's product number 3, because remember we're going from pyruvate plus NAD plus CoA goes to acetyl-CoA plus NADH plus a CO2. So everything else in between, the TPP, the lipoamide, the FAD, those all get regenerated in the enzyme. Okay, so a couple other things that we're going to talk about. So TPP is created from vitamin B1, which is also known as thiamine. Let's see here where am I gonna go next? Let's go to CoA. So CoA comes from vitamin B5, which is also known as pantothenic acid. Tothenic acid. FAD comes from vitamin B2, which is also known as riboflavin. That's an R. We've already met NADH and its vitamin before. And then the last one isn't necessarily from a vitamin. You might think of it as like a supplement, like another molecule that you need to eat. But it comes from lipoic acid. Okay, and the reason that lipoamide is called the swinging arm is because it has this really, really long structure, and it will pick up, I don't know how to do this without drawing over this whole structure. It will pick up the two carbons in E1 and swing them over to E3. And I think I've added an animation that I found online that I think is really cool. I think I've added it to the course website. So please check that out so you can see this goofy looking swinging arm. But there's another swinging arm that we'll meet later. So that is, that's the PDC, or the pyruvate dehydrogenase complex, 3 subunits, 5 coenzymes. We've just met 4 new vitamins and supplements. I don't know, supplement's not quite the right word. Extra molecule you need to take. And now we got to move forward to the TCA cycle. But before we do that, we need to talk about this really interesting thioester bond, because I think you've heard of acetyl-CoA before, and there's a reason for that. It's a very central carbon intermediate in a number of metabolic pathways. Okay, so the thioester bond just put CoA on here. The thioester bond is this one, and hydrolysis of it yields about a delta G that's about negative 31 kilojoules per mole. So creating the thioester bond obviously costs a lot of energy, but hydrolysis of it will release a lot of energy. So oftentimes this thioester bond is created just to set up a future coupling reaction. Okay, so one of the next reactions that we're going to see is going to be the first step in a new pathway, and those are typically irreversible, and we're going to break the thioester bond in order to facilitate the irreversibility of that reaction. But before we do that, I'm going to go through the different structures of the coenzymes and their vitamins so that you've seen them all once. Okay, so the E1 subunit contains a coenzyme thiamine pyrophosphate. The two phosphates together, this is what's called the pyrophosphate. And the way that I recognize thiamine pyrophosphate, one is it has 2 of those, and the other one is this 5-membered ring that has a sulfur and a carbon and a nitrogen all in a row, and that's pretty unique. And like I said, this comes from vitamin B1, which is thiamine, so that's the precursor. So hopefully you can again see here is the nitrogen, the carbon, and the sulfur all sandwiched in there together. And thiamine is the precursor for TPP in E1, and it's found in a variety of foods, including meats and grains. Lipoamide is the swinging arm, and it comes from lipoic acid. Oh, there it is. And the reason it's the swinging arm is it's a really, really long structure that can reach between E1 and then swing to E2 and then swing to E3. You'll notice that this is why it's called lipoamide, because it has an amide group in it, and hopefully you can also see that it's coming is from the side chain of a lysine or a lysyl. So the side chain of a lysine will create a peptide bond or an amide bond with that carboxylic acid. You've seen that mechanism before. It's the same thing as peptide bond synthesis. And yeah, that's lipoic acid. So be able to recognize that. How I recognize it, it's a 5-membered ring with 2 sulfurs in it. And then that gets us also to the second one that's an E2, which is coenzyme A, and this activates acyl groups. In our case, it's an acetyl group, which means it only has 1 carbon. Acyl is more generic, and it can be many, many, many carbons. Again, acetyl-CoA is humongous. There's a nucleotide here. Sorry, I should have grabbed that piece. So we have a base, a sugar, and a phosphate. So there's a nucleotide. There's a whole bunch of, just a lot going on in the middle here. But the ending piece is a thiol, an SH. And what it creates are these thioester bonds, where regardless if it's an acetyl or an acyl-CoA. And we'll meet some acyl-CoAs when we talk about lipids, lipid metabolism. Okay, again, just to recognize this, only part of this is pantothenic acid, which is, again, one of your vitamins. So, again, pantothenic acid, B5, acetyl-CoA is just this little teeny tiny functional group that gets added to the end of it. But just be able to recognize it. Some unique things that I see about this is there's a nucleotide in it, a whole bunch of crazy, and then it always ends in an SH. Vitamin B2, which is riboflavin, now we're into enzyme subunit E3 of the PDC, and this is our second reduced coenzyme that will also transport electrons. So it's sort of analogous to NADH. Let's see. So FAD stands for flavin adenine dinucleotide. So that means it again has 2 nucleotides. Here's one. Here's the other one. Just like nicotinic adenine dinucleotide, they both have nucleotides in them, but the riboflavin has this unique 3-ring structure. So there's this interesting 3-ring structure that makes FAD different from NAD+, and FADH2 is a 2-electron, 1 hydrogen carrier. If you remember that NADH is also a 2-electron carrier, which makes sense. There's 2 electrons in a covalent bond, but it's only a 1 hydrogen carrier, because that's why we go from NAD+ to NADH, not 2 hydrogens, just 1. In here, we're going to grab 2 hydrogens, so you get to have FADH2. Anyways, again, just be able to recognize this structure. Okay, so let's recap here just a little bit. So we'll do, I like to repeat this drawing, so you'll see it a whole bunch. So this will be the blood or extracellular with some glucose on the outside, and then a membrane transporter permease doing facilitated diffusion to get glucose into the cell. This is the cytosol. Again, this is a super zoomed in view. And then our glucose will undergo glycolysis. I don't know why there's 2 lines there. Let's back this up. Hey, come back here. Okay. We'll undergo glycolysis to form 2 pyruvates. In the process, we get NADH out, and we get ATP, 2 of those, 2 of those. Some of the pyruvate will be made into lactic acid. I'm just going to put La here, and the purpose of that is to regenerate the NAD+ to keep glycolysis going. Now, another chunk of this pyruvate will enter into the mitochondrial matrix. So here's a crazy, super zoomed in view of the mitochondria. It is not as big as the whole cell, but this is going to be the outer membrane, so I'm going to put OM. This second line here is going to be the inner membrane. And the space in between is the intermembrane space. And then all the way on the inside is the matrix. Okay. The outer membrane of the mitochondria is fairly permeable and not very selective, so it's the inner membrane where you'll find most, if not all, of your transport proteins. So in order to get pyruvate in, it will cross the outer membrane, not very selective, but it will need a transporter to get through the inner membrane because it is small, it's polar, and it's charged. And then once it gets in, it will undergo a little bit of oxidation to make acetyl CoA, plus we'll get some CO2 will come out as well as NADH. And again, this is the PDC reaction. Okay, so we're in the matrix. Our 3 carbons from pyruvate are now into 2 carbons and 1 carbon. And the TCA cycle is going to complete the oxidation of these pyruvate molecules. So we should be getting 2 CO2s out when we do this. All right, here we go. Let's see. I'm just going to draw it all here. All right, so we're going to start with OAA, which is called oxaloacetate. And this is a 4-carbon molecule. And it's actually the last molecule in this cyclic cycle. But the reason we're starting with it is because the last becomes sort of the first. So this 4-carbon molecule will combine with a 2-carbon molecule to make something that is 6 carbons. 4 plus 2 is 6, and that's called, whoops, I can't see that. It's called citrate. Okay, and citrate has 6 carbons, and if you'll notice, we lost our CoA. All right, so this reaction is irreversible because of that thioester bond hydrolysis releases a lot of energy, and the enzyme that catalyzes this irreversible step is called citrate synthase, because it creates citrate. Okay, and then the next step is a reversible one, and that creates isocitrate. Again, 6 carbons here because we haven't lost any CO2s yet. And then the third step is another irreversible, and that creates a molecule called alpha ketoglutarate. Okay, also known as alpha KG. That's the abbreviation. There aren't very many abbreviations for the TCA cycle like there were for glycolysis. There's a couple, but not nearly as many. Here's where we're going to lose our first CO2. So that means that alpha ketoglutarate must have 5 carbons because we lost 1 CO2, and a CO2 has only 1 carbon in it. We're also going to generate our first NADH, so hopefully that makes sense to you. And the enzyme that catalyzes this reaction is called isocitrate. And I'm going to put a DH for dehydrogenase. So all the dehydrogenases that will meet from here forward are all named for their substrate. So because this one takes in isocitrate and it does a redox reaction to create alpha ketoglutarate, it's an isocitrate dehydrogenase. And then the next step is our last and third irreversible, and that takes alpha ketoglutarate to a molecule called succinyl-CoA. Succinyl-CoA has 4 carbons, which means that you know that we lost another CO2. We gained an NADH. And just note that when I write an arrow that an NADH is coming out, an NAD plus had to go in. I'm just trying to save a little bit of space here. We added on a CoA, so we need to add a CoA here going in. And these arrows coming off of the These arrows coming off of the arrow, or the arrow is going out. Oh, goodness gracious. Okay, and then this enzyme, because again, there's a redox reaction going on here, it's alpha ketoglutarate dehydrogenase. Again, the dehydrogenase is named for its substrate, alpha ketoglutarate. Okay, from here, we've created another thioester bond, which means it's been created in order to set up another reaction that needs to be coupled. So we go from succinyl-CoA to succinate. Again, 4 carbons here. Everything from here forward is going to be 4 carbons. If you'll notice, oxaloacetate is 4 carbons. It's the last thing. So we've lost both of our CO2s, which is exactly what we wanted. We wanted 2 CO2s because we put 2 carbons into it. Succinyl-CoA to succinate will generate a GTP from a GDP and a PI, and that's going to cost energy. It's going to cost a lot of energy, and the payment for that energy is the thioester bond cleavage again. Then we'll go from succinate to fumarate, and here we're going to get out an FADH2, and then we'll go from fumarate to malate, MAL, and then malate to oxaloacetate, and we get our third and last NADH. Okay, so let's look at what we have. So we had 2 carbons going in. We have 2 CO2s coming out. One right here, one right here. We get 1, 2, 3 NADHs. And we get 1 FADH2. And we get 1 GTP. And GTP, just so you know, is the equivalent of an ATP. So anytime you get a GTP, you've essentially made ATP. What you'll notice is that we didn't actually make a whole ton of ATP. That's because these reduced electron carriers are going to eventually go to electron transport chain to help generate ATP through oxidative phosphorylation. But let's go back and take stock just one more time. Glucose has 6 carbons. So when all is said and done, we have to have 6 CO2s. Okay, just roll with me on this one. It goes to 2 pyruvates, and each of those has 3 carbons. Both of those, well, let's just say both of those pyruvates go into the matrix. So I get 2 pyruvates going in here, so I should have 2 CO2s there. So I'm going to collect 2 CO2s after my PDC. I have 2 acetyl-CoAs. If I get 2 pyruvates in, I have to have 2 acetyl-CoAs coming out, which means that this whole cycle is going to have to spin 2 times in order for me to get 4 CO2s out. And this is how I get 1, 2, 3, 4, 5, 6, and those are all 6 of my CO2s that I get from glucose. So I've gone from glucose and completely oxidized it to 6 CO2s. Now, what you'll notice is that we again have put in no oxygen. And in order to completely catabolize this, we have to add oxygen and water, and eventually we'll get there when we do the electron transport chain. But so far, we again have not added in any O2, but we have completely catabolized. Sorry, I'm just checking my notes. We've completely catabolized or completely oxidized the glucose. We've taken every CH bond in glucose and turned it into CO bonds. Phew. Oh, hey, look, let's fill in a diagram. I like this one a lot. Okay, all the boxes in the middle are for a number of carbons. So what color do I want? Okay, so acetyl-CoA up here has 2 carbons. Oxaloacetate has 4 carbons. I like this diagram because you actually get to see the molecules. I don't know if you have any interest in them, but you'll notice there's 1, 2, 3, 4 carbons there. Citrate then has to have 6 carbons, and it does. 1, 2, 3, 4, 5, 6. And then none of these arrows, this one should be like a double-headed arrow. This one should be double, 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 double. These ones don't really show the irreversible steps that well. We don't lose any CO2s when we go to isocitrate, so that also has 6 carbons. We do lose a CO2 from isocitrate to alpha-ketoglutarate, which must be that little bubble right there. And we do get an NADH out of this, so we go from NAD plus to NADH, and that leaves us with alpha-ketoglutarate, which is 5 carbons. Again, we go from alpha-ketoglutarate to succinyl-CoA, and we lose another CO2, which means succinyl-CoA is 4 carbons. We get another NADH out here. And then we also need to put in a CoA. That's what this guy is right here. That's a CoA going in, obviously. How is that obvious? It's not obvious. Yikes. This one's a little bit harder to fill in. And then everything from here forward has to have 4 carbons. So we get back to our 4-carbon oxaloacetate. Going from succinyl-CoA to succinate, we get a CoA back out, but and that enables us, that thioester bond cleavage enables us to push together a GDP with a inorganic phosphate to create GTP. And then succinate to fumarate, we get an FA. Whoa, what just happened? It moved. The whole thing moved. Why? FADH2 from an FAD. And then there's a, from fumarate to malate, nothing really happens. It's a rearrangement of the atoms. And then from malate to oxaloacetate, NAD plus to NADH. And again, we're getting NADH out here, so that means there's an oxidation reduction. So there's malate dehydrogenase. It's not one of the ones that you need to know, but hopefully it makes sense. This one is succinate dehydrogenase. Again, these are all named. All the dehydrogenases, the ones that create these reduced coenzymes, are named for their substrates. The rate-limiting step is isocitrate dehydrogenase. It turns out in the TCA cycle, it could be citrate synthase, or it could be alpha-ketoglutarate dehydrogenase, but more often than not, people say that it's isocitrate dehydrogenase. So we're going to go with that one, and I missed one bubble right here, and that is the CoA that comes out, that comes off of the acetyl-CoA for citrate synthase. And again, you'll notice that in this pathway, the very first step is irreversible, just like in glycolysis. This serves a number of purposes. It keeps the citrate concentration high, so it pushes the reaction forward. And because this is cyclic, it keeps oxaloacetate concentration low and pulls all of these irreversibles again forward. So remember, all of our pathways have to proceed in a forward exergonic direction, and the TCA cycle is no exception to this. Okay, so this is just kind of like an overall big picture recap. So 1 glucose to 2 pyruvates. We got 2 ATPs, 2 NADHs. Then we have our bridge step that goes from pyruvate to acetyl-CoA. Again, if we get 2 pyruvates here, we get 2 NADHs out, 2 CO2s. And then that acetyl-CoA goes into the TCA cycle, and it again will go around 2 times. So we get 6 of these. QH2 is sort of the same thing as FADH2. 2 GTPs and 4 CO2s. So remember, 4 plus 2 gets us back to 6 carbons, and hopefully you're following my weird biochemistry carbon math. I'll see you later. That's all I have. Bye-bye.